Hello, my name is Professor Hudson, and you're about to view narrated PowerPoint lectures for Chapter 11 on Mendelian genetics. In the last segment, you were given a practice problem to see if you could set up a Punnett Square in order to determine if two parents with unattached earlobes, what are the chances of them also having a child with unattached earlobes? Let's take a look at how you would work this problem. First thing you have to do is identify the genotypes of what the parents would be. In the question, they tell you that the parents are heterozygous for unattached earlobes. Remember, heterozygous means one of each version of alleles, one dominant and one recessive. So for the male parent, using the letter E for shorthand, we write a capital E and a lowercase e. For the female parent, we write a capital E and a lowercase e. This designates that both parents are heterozygous. Then, based on what the parents' diploid heterozygous condition, write down the possible gametes that each parent can make. Remember, Mendel's law of segregation states that if an organism is diploid, when they produce sperm and eggs, the sperm and egg will only receive half or become haploid due to segregation of the alleles. That means that this heterozygous male will make sperm that either have the dominant version or the recessive version. There will not be both in his sperm. It will be one or the other. The same thing for the female. When she makes eggs, the eggs will either have a dominant version of this trait or a recessive, not both. Remember, the sperm and egg are haploid, so they must have half of whatever is the diploid condition. Once we know what the sperm and eggs are, we write a Punnett square. Typically, with monohybrid crosses, this Punnett square will have 4 boxes. However, you can figure out how many boxes to make for a Punnett square for different type of crosses based on how many different eggs and sperm you have. In this case, we had 2 different types of eggs that could be made and 2 different types of sperm. 2 times 2 is 4, so that tells us there's going to be 4 possible combinations. Therefore, I need 4 boxes. I put the male gametes on one side of the Punnett square. In this case, they're on the top. Then I put the female possible sex gametes on the side. Then I do a cross. I pretend that the sperm has fertilized the egg and write down the possible combinations. For example, if a dominant sperm with a dominant allele for the earlobe gene fertilizes a dominant egg with the dominant version of this earlobe gene in it, then the offspring will be homozygous dominant. Remember, the sperm and egg are haploid, but when they fertilize, you get the diploid back. So in this first box, we have a homozygous dominant. In the second box at the top, the sperm had a recessive allele and it fertilized a dominant allele in the egg. In this case, the recessive and the dominant make a heterozygous for this trait. Then in the bottom row, we must have had a sperm with a dominant allele fertilizing an egg with a recessive allele. This gave us the heterozygous. And in the last box at the bottom, you have a recessive sperm with its recessive allele fertilizing an egg with its recessive allele to get the homozygous recessive. Now typically when you do a Punnett square, the question will ask you to figure out the genotypic and phenotypic ratios along with the genotypes and phenotypes. The genotypes are given by the letters in the Punnett square. The genotypic ratio is how many times out of 4 do we see the different combinations of these genotype in the offspring. So the genotypic ratio in this case is 1 out of 4 with the homozygous dominant given by the 2 capital E's, 2 out of 4 with the heterozygous, meaning 1 dominant and one recessive, given by the capital E and the lowercase e, and one homozygous recessive, given by the two lowercase letters. The genotypic ratio is 1 to 2 to 1, or 1 out of 4 to 2 out of 4 to 1 out of 4. The phenotypic ratio is where you discuss the phenotypes that you see and what their ratios are. In this case, the possible phenotypes are unattached earlobes and attached earlobes. In this case, there's a 3 out of 4 chance of having unattached earlobes, given by the homozygous dominant and the heterozygous conditions, and a 1 out of 4 chance of being homozygous recessive and having attached earlobes. This Punnett square actually shows what we just did. Notice here that with two heterozygous parents each having the unattached earlobes get together, the chances of having unattached earlobes in their offspring is 3 out of 4, or 75%. And there's a 25% chance, or a 1 out of 4 chance of having attached earlobes. Note, this is a situation where you have two parents that have one phenotype, but when they have children, some of the children may have a phenotype that you don't see in either parent. This is because certain phenotypes are associated with recessive alleles, and the recessive will only come out if you get one from each parent. Note that the parents had unattached earlobes, yet each one had to have been carrying a recessive allele for the attached earlobes, and this recessive allele came out when the child received one from each parent. Here's another cross that you can try on your own. Directions. Using the results of a Punnett square, answer the question below. Widow's peak is dominant to straight hairline. If the parents are heterozygous for widow's peak, then what are the chances of having a child with a straight hairline? Take a moment and answer this question. You can pause the video here, and then after you've had a chance to try drawing the Punnett square on your own, the next slide will show you how it would be set up. Here is a Punnett square showing the results of the cross on the previous slide. When the parents are heterozygous, as they were in the question, both having a widow's peak, but heterozygous for their widow peak, note that each parent can produce sex cells that have the widow's peak gene or the straight hairline gene. In the offspring, there will be a 3 out of 4, or 75% chance of having a widow's peak as the phenotypic ratio, and a 1 out of 4 chance of having a straight hairline. Just as an aside, when doing Punnett squares on your own, try to use letters where the upper and lower case do not look the same as is in this example. Sometimes if you use the upper and lower case that look the same, such as in the case of W or S or P, it can be confusing as to what is the dominant and what is the recessive. Now that we've talked about monohybrid crosses and how to set those up, let's talk about how multiple traits are inherited and how you would set up a Punnett square if you were looking at more than one trait at a time. In the previous examples, we only looked at one trait per cross. In multiple trait crosses, you can look at more than one trait at a time. However, the more traits you look at in one Punnett square, the bigger the Punnett square will be. So how are multiple traits inherited? Mendel concluded the origination of single traits of inheritance. He then pursued more complex questions relative to inheritance of multiple traits. His initial experience included crossbreeding plants that differed in two traits. For example, he looked at seed color, such as yellow or green, and seed shape, smooth or wrinkled, in the same Punnett Square cross. So he was no longer looking at one trait at a time. He moved on to looking at inheritance of two traits at a time. When you look at two traits at a time in a single cross, we call that a dihybrid cross. Notice that in this case, he looked at the inheritance patterns in between a yellow, smooth seed crossing with another yellow, smooth seed. And notice, since he was looking at two traits at one time, and each trait was heterozygous, there were actually 4 different possible sperm and 4 different possible eggs. So for that reason, 4 times 4 is 16. And so he needed to draw a Punnett square that had 16 boxes. As an aside, whenever you cross a double heterozygous for both traits with another double heterozygous in a 2-trait cross, you should get a ratio of phenotypes that reads 9, 3, 3, 1, or 9 out of 16, 3 out of 16, 3 out of 16, or 1 out of 16. This only occurs when the cross being done is a dihybrid cross with heterozygous at both traits. The 9 out of 16 is the double dominant condition, and the 1 out of 16 is the double recessive condition. Based on his work with the dihybrid crosses, Mendel developed another law. This is called Mendel's law of independent assortment. It states that random alignment of chromosome pairs during meiosis I leads to the independent assortment of alleles found on different chromosomes. The independent inheritance of 2 or more traits is called the law of independent assortment. Multiple traits are inherited independently because the alleles of one gene are distributed to gametes independently of the alleles for other genes. Independent assortment will occur when the traits being studied are controlled by genes on different pairs of homologous chromosomes. The physical basis of independent assortment has to do with the way homologous pairs line up during meiosis. Which of the 2 chromosome homologous pairs is on top occurs randomly for all pairs. So the homologous chromosomes assort randomly and independent of one another at anaphase 1. Let's look at an animation that illustrates Mendel's law of independent assortment.
During metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
This slide shows another example of a dihybrid cross. In this instance, the traits being crossed are hairline and finger length. Note 16 boxes were needed this time because in the F1 generation you had individuals that were heterozygous for both traits. When they were crossed, you ended up with the 9:3:1 ratio of the phenotypes. Now in the next segment, we will talk about rules of inheritance that don't follow traditional Mendelian patterns. According to Mendel, each trait is completely controlled by a single gene, only 2 possible alleles of each gene exist, and one allele is completely dominant to the other, which is the recessive allele. In the next segment, we will see situations where this is not always the case.